Does it show up on YouTube, Amanda?
It is live streaming now.
Yeah.
Okay, so everyone out there watching, we are doing the same thing as the previous session. We're live streaming on YouTube and we're posting a link on Facebook. So give us just a minute to get all of that done. And please take a look at our slides for our sponsors.
It's my pleasure to introduce Katie Angione, genetic counselor from Children's Hospital. As she's talking, please send your questions our way so that we can learn with her. She's a dedicated genetic counselor and willing to answer your questions as they come in at the end of her talk. With that, I will hand it over to Katie.
Awesome. I want to verify real quick. Amanda, are we on Facebook as well? Or do we need to wait a minute? Amanda, are you on mute?
I am on mute and the link should be coming through to Facebook anytime.
So questions can go to Facebook, to the questions at CDKL5.com, and also on the chat in the live YouTube video.
And if there's questions specifically for me, I'm happy to answer them. Dr. Benke is going to be giving a talk right after me, and we're going to both stick around afterwards to do sort of a joint Q&A. So if you have questions just for me, that's fine. If not, you'll have another opportunity after Dr. Benke's talk.
Thank you. And I'll say one more thing. So we did do a formal welcome earlier on the first session. And so if you missed that session, please, take some time, check it out. Dr. Jenny Downs did a great job talking about quality of life and giving some ideas on how to improve that. And with that being said, we can begin.
All right, perfect. All right. Is everyone able to see that?
Yes.
Okay. All right, so I'm just going to turn off my video so I'm not being distracting. So a lot of you might have already seen the Genetics 101 talk from back in 2018, where we went over some of the basics of genetics. We talked about different types of variants, touched on some genetic concepts. So the purpose of today's talk is to just take a deeper dive into a couple of those genetic concepts. So I'm not going to spend too much time on the basics. I definitely invite anyone to go back and view that previous webinar if you want sort of a refresher, but this one's going to be specifically focusing on a couple of those concepts. There we go. Nothing to disclose. So today we're going to talk a little bit more about X-linked disorders and X-inactivation. We're going to talk about somatic and germline mosaicism and how that applies to differences in CDKL5 deficiency disorder. And we'll go over a couple of specific types of genetic mutations that are targeted by two of the potential CDD treatments. So I'm not going to go over CDKL5 deficiency disorder. You all are very familiar with the symptoms caused by it. But we know that all of these symptoms are caused by a single genetic change in the CDKL5 gene. So when we say a genetic change, we're talking about any variation in that gene. And in terms of CDKL5 deficiency disorder or any disorder, we're talking about changes that impact the function of that gene. So we might refer to these as pathogenic variants or genetic mutations. So I'll sort of use all of those terms interchangeably. The CDKL5 gene is made up of 22 exons, and exons are more or less just the parts of the genetic material that make up the final CDKL5 protein product. There are a few different versions of that final product. So in this picture, the part in green is sort of showing the version of the CDKL5 protein that is expressed in the brain, and that's usually the version that we're most concerned about in terms of CDKL5 deficiency disorder. CDKL5 is located on the X chromosome. This is just a map of what the X chromosome looks like, and CDKL5 is located right here. And that's really important for how this gene functions and the differences that we see between males and females with the condition. So chromosomes are basically packages of genetic material, kind of like the chapters in a book or volumes in an encyclopedia. And biological males and biological females, for the most part, have the same chromosomes, with the exception of the sex chromosomes. So that's the X and Y in a male and 2 X's in a female. So the fact that females have 2 X chromosomes is the reason that most patients with CDD are female. So at this point, we think that number is between 80 and 90% of people with CDD are females. And because females have a second X chromosome, they have a second copy of the CDKL5 gene that is usually typical or normal. So while their bodies aren't making enough of the CDKL5 protein, they are at least making some of it. Males, however, don't have that second, oh, sorry, was there? Oh. So, sorry, I lost my train of thought. So males don't have that second copy of the gene, so they might be more severely affected. And there's an asterisk there, we'll come back to that. We're actually learning that that might not actually be the case. But some males are more severely affected. Some males actually probably don't survive to term because there's only one copy of that gene and it's not working correctly. So because females have twice the genetic material on the X chromosome as males, one of the 2 X chromosomes is actually turned off in each cell. So this is something called Xinactivation. So Xinactivation ensures that females, like males, have one functional copy of the X chromosome in each cell. So that sort of gives females the correct dosage of all of the genetic material that lives on the X chromosome. So most of the time, this process is completely random. So in about half of the cells, the one X is turned off, and in the other half, the other X is turned off. So this leads to a picture sort of like this, where you have some copies or some cells that have one copy of the X chromosome turned on and other cells that have the other copy turned on. But sometimes this doesn't happen in a completely random way, and it might be skewed in one direction or another. So we might end up with more cells that have one copy of the X turned off than the other. So this is important in X-linked disorders because that can affect how severe or how less severe that condition might be, depending on which direction that X inactivation is skewed. So a really good example of this is seen in tortoiseshell cats. So the gene that decides whether their fur is orange or black is found on the X chromosome. So a female cat might have one copy of the gene that has the code for orange and one copy that has the code for black. So the gene that's active in a particular group of cells is going to determine what color that patch of fur will be. So the black patches are going to come from groups of cells where the black X is active and the orange is inactive. And the orange patches are going to come from groups of cells where the orange copy of the gene is active. So this kind of results in this random pattern where you see patches of orange and patches of black sort of randomly dispersed. But when that X inactivation is skewed, you might end up with a cat that's mostly orange or a cat that's mostly black, depending on which gene is being expressed in the majority of the cells. So obviously, when we're talking about cats, this doesn't really, it's not really a big deal. It doesn't really make a difference. Those different genes are just causing different colors. But when we're talking about an X-linked disorder like CDKL5, the proportion of typical versus mutated CDKL5 genes can have a really big effect on the clinical outcome of that patient. So depending on the proportion of typical genes being expressed versus genes that have that mutation, that person's symptoms might be more mild or more severe. So in theory, someone with highly skewed X inactivation, and I'll just go back to that other slide. So someone with highly skewed X inactivation that's favoring the typical or normal copy of the CDKL5 gene might have very mild symptoms. They might have no apparent symptoms. So this is something that that we've seen in other conditions like Rett syndrome, where a mother might have the mutation, but she might have highly skewed X inactivation. So she's not actually expressing the mutation in the majority of her cells. So this isn't something we've seen yet in CDKL5, but it is definitely theoretically possible. So one of the important things about kind of understanding X inactivation as CDD is that it could be a potential treatment target. So there are actually a subset of genes that escape the process of X inactivation. So in reality, the entire second copy of that X chromosome is not being turned off all the way. There are some genes that remain active. And those genes that remain active have some genetic markers in common. So the thought with this as a potential therapy is that if we could find a way to create or apply those same markers to the CDKL5 gene, we might be able to sort of rescue that inactivated normal copy. So this is something that Kyle Fink's lab is working on at UC Davis. So it's something that I think has a lot of potential. There are still a lot of hurdles to jump in making this a realistic treatment option. So there's a lot of steps we need to overcome to get to that end goal of using this as an actual treatment. At this point, Kyle Fink's lab has figured out how to target the CDKL5 gene in culture. But then we need to move on to figuring out how to do that in mice, figuring out how to do it in humans, and then making sure that it'd be something that's being spread throughout the body, especially in the brain. So if we're fixing this or rescuing that inactivated normal copy of the gene in most of the body, but not in the brain, we're not going to get that impact that we're looking for, because the brain is what's really important to make sure that we're affecting the cells that are in the brain and that are causing the majority of symptoms that we see in CDD. So the bottom line here is that we do have a long way to go, but you have to start somewhere. So I think that this is definitely a good potential future treatment option. It is also important to note that this type of treatment would only be possible for females, because males don't have that second copy of the gene to reactivate. So this is something that might be a potential therapy for a good portion of CDKL5 patients, but definitely not everyone. So another important genetic concept that plays a really big role in CDD is mosaicism. So in mosaic artwork like this, you have many different tiles of different colors and shapes that make up one image. In genetic mosaicism, instead of someone having the exact same genetic material in every cell in their body, there are some cells that have a genetic mutation and some cells that don't. So mosaicism happens when there is a random change or a mutation that arises very early on in embryonic development. So you can see this is the initial cell and it's normal. There's no mutation there. So in mosaicism, the genetic changes are not present in the fertilized egg. They're not present or passed down from parents, but they happen later on in development. So as the cells continue to divide, cells that arise from the cell that has the altered gene are going to have that mutation, whereas the other cells will not. So depending on the timing of that first mutation, that kind of determines the proportion of cells that will have the mutation and the proportion that have the normal copy of that gene. The timing is also going to determine which tissues may or may not be affected, because there's different tissues that sort of form at different stages of development. So until recently, it's been pretty difficult to detect mosaicism through genetic testing, but over the past several years, there's been a lot of advancements in technology that have greatly improved our ability to detect mosaicism. So now we are starting to understand that it's actually a bit more common than we originally thought it was. So this is a paper in 2018 that explored rates of mosaicism in 9 different epilepsy genes, including CDKL5. There were 91 CDKL5 patients in this group, and overall there was a mosaic rate of about 9% in those patients. You can see that rate is much higher in males. There's a rate of about 32%. And this is not just present in this paper. There are kind of larger clinical cohorts that are showing similar rates of mosaicism in males. And in females, it's much less common for a mosaic mutation to be present. So there are a lot of challenges in trying to assess what effect mosaicism will have on the clinical outcome of any given CDD patient. So first off, while technology has come a long way, we're still not able to predict the precise level of mosaicism in a sample. So labs will typically report something like possible mosaicism or suspected mosaicism. They're not going to quote an exact percentage or even a range of percentages in most cases. That being said, even if we were able to determine the exact level of mosaicism in a tissue, the level of mosaicism in the sample that we're actually testing, which is usually blood or saliva, doesn't necessarily reflect the level in the brain. And for a neurodevelopmental disorder like CDD, the level in the brain is what would be the most relevant. There's also some challenges in understanding exactly what effects mosaicism might have. And those are kind of different in different genders. So in females, it's possible that there might be a couple of different things going on. So a female might have a mosaic mutation. It's also possible that they have skewed X inactivation as well. So it can be kind of tricky to tease out which of those might be affecting their clinical presentation. And then in males, it's just been really difficult to study any phenotypic differences because we're working with a much smaller sample size. We don't have huge numbers of patients in our clinical databases to begin with. We've definitely come a long way in getting patients enrolled in those groups, but we know there's a lot more patients out there than we have represented. And if only 10 or 20% of those patients are males, and then only a third of those males are mosaic, that's a really small number of patients. So we just need to be careful about drawing any conclusions based on, that small population size. And I kind of touched on this earlier, but years ago, we thought that males with CDD were more severely affected than females. But we're starting to see with more recent data that there's not really a big clinical difference between the genders. So one reason for this might be because years ago, testing was not able to detect mosaic mutations. So males that had mosaicism might not have been picked up on testing years ago. So it could be that, we're just starting to learn more about those patients. And we're learning that, there's more males that might be more mildly affected because they have a mosaic mutation. So, hopefully as we're able to extend research and get more numbers of patients enrolled, we're going to be able to answer some of these questions a little bit better. For what it's worth, I will say that the mosaic males I have seen in clinic have definitely been less severely affected than the average CDD patient. And we can't talk about mosaicism without talking about germline mosaicism. So this is when there's a mutation present only in a portion of the germ cells, which is the eggs or the sperm, but not present in the rest of the body. So this is important because germline mosaicism is not going to be picked up on carrier testing on a blood or saliva sample. So we're not able to test parents and see if there is a germline mosaic variant. So far, there have been 2 reported families with CDD who have a germline mosaic variant. So both of those families actually had, I think they both have 2 daughters. So each family had a second child with CDD because there was a germline mosaic variant present. And unfortunately, it's just really hard to know that until a pregnancy is already, in progress. Aside from germline mosaicism, there's a couple other scenarios where there might be a recurrence risk for CDD, so the chance of having another child who's affected in the same family. So this could happen if a parent had low-level mosaicism throughout their body, so somatic mosaicism, that extended to their germ cells. So this is termed gonosomal mosaicism, so basically mosaicism in both the body and in the germ cells. Carrier testing might be able to pick this up in the parent depending on the proportion of cells with the mutation. If that were the case, we would be able to at least counsel the family, let them know that there is an increased chance of recurrence, and then offer any prenatal genetic testing or prenatal counseling for future pregnancies if that was something they wanted. Carrier testing would also pick up a variant present in a mother that was not expressed due to highly skewed X inactivation. So again, this would allow us to offer prenatal counseling and testing options to that family. We haven't seen this, and I think I mentioned this before, we haven't seen this in CDD so far, but we have seen it in Rett syndrome. So we definitely think that it is a possibility. And that's one of the reasons that we do recommend parental testing, or at least testing of the mother for families who are wanting to have more children and wanting to know if there is a chance of having another affected child. So I just want to pause for a second and see if there's any immediate questions so far. If Karen or Heidi can let me know.
There haven't been any immediate questions coming in about mosaicism or X inactivation. So I think you can continue. Maybe by the end we'll have some.
Okay, sounds good. So I just wanted to go over a couple of the therapies. Oh, sorry, I've got a fire truck going by my window. The joys of working from home. So I wanted to go over a couple of therapies for CDD. So I wanted to talk about both of these specifically because they target a specific type of genetic variant. So Adalorin uses something called read-through therapy. So this is a drug that I'm sure many of you are aware of. It's in phase 2 clinical trials. And this is going to target nonsense mutations in the CDKL5 gene. So nonsense mutations, some of you might remember this slide from the previous talk. A nonsense mutation is any change in DNA that signals the cell to stop building the protein too early. And usually this causes a shortened protein or an early truncation. So the gene is basically going to stop short and not create a complete protein. So in the analogy here, you can see the sentence, the cat ate the rat. If there's a nonsense mutation at this letter A, you would cut off that sentence and you wouldn't know what happens at the end of that sentence. So Adalorin basically works by forcing the protein coding machinery of the cell to just ignore that premature stop signal and continue reading the rest of the gene in order to produce the full length protein. So if this worked perfectly, it would make a lot of sense. Unfortunately, there are some imperfections to the process. So number one, there are multiple different signals that tell the cell to stop building the protein. So there's not just one particular code that introduces this early stop codon. There's a couple different variations of that code. And some of those signals or some of those codes respond better to this type of therapy than others. There's also differences in how the codes surrounding those stop signals might influence how effective this type of therapy is. So the bottom line there is that some nonsense mutations might be more amenable than others to this type of treatment. So having a nonsense mutation alone does not guarantee that this would be an effective treatment. Read-through also increases the chance that a new missense mutation will be introduced. So using this therapy, you might run the risk of fixing one problem and creating another. So this might limit which nonsense mutations can be targeted. So this is kind of a schematic of what the spread of mutations looks like across the CDKL5 gene. And in green, those are missense variants where only one, it's kind of a typo, one letter is changed for another. And then in blue, these are truncation variants. So a lot of these are nonsense mutations that are causing an early truncation. So you can see that the missense variants are all sort of grouped in this area here, whereas the nonsense mutations might be more spread out. But it might only be a good option, this type of therapy might only be a good option for nonsense mutations that are outside of this region, because if we introduce a missense mutation in this region, what we know about those mutations so far suggests that if a missense mutation happens within this area of the gene, it's more likely to cause disease. So we want to be really careful about taking the risk of creating a missense mutation in this area. So basically, it might make more sense to target nonsense mutations that are outside of that area. And then lastly, the rescued version of the protein usually doesn't work quite as well as a typical CDKL5 protein. So, we might be able to rescue the protein, but it might not fully restore the function. So there still might be less active or less functional CDKL5 protein than we need. So another really exciting therapeutic is antisense oligonucleotide, I can never say this, ASO, antisense oligonucleotide therapy. And this is a really hot topic in the past few years. It's been used for a lot of really exciting drugs and other neurologic disorders. So including Spinraza, which has been used to treat thousands at this point, thousands of patients with spinal muscular atrophy. It's also the mechanism for Melicin, which was the drug designed for Mila, a single patient with CLN7 Batten disease. So ASOs can actually work in a couple of different ways, but the method that's really relevant to CDKL5 is acting on splice site variants. So splicing is the process where the body removes the introns, so these regions of the gene, and puts together the exons, which are the part of the gene that actually code for protein. So there's sort of this process of cutting up and putting back together these different portions of the gene in order to end up with that final product that's going to code for the functional CDKL5 protein. So splicing can actually happen in a lot of different ways, and the body naturally uses a lot of these different ways in any one gene, but there's usually sort of a primary, for lack of a better word, correct version of how splicing occurs. So in a healthy cell, that correct version is what's being made the majority of the time. But a splice mutation causes one of these alternative events to happen too often. And that usually leads to less production of the correct protein and more production of this abnormal version of the protein. So that abnormal version might not work properly. It might get broken down by the body. So either it's getting in the way by creating this abnormal version of the protein, or it's just making the entire process less efficient so that there's less of that normal protein being made. So in Spinraza, the ASO works by actually encouraging the body to incorporate an exon of a gene called SMN2. So SMN2 is sort of a backup for the main gene that's implicated in SMA, which is SMN1. So my colleague Melissa usually describes it as a big brother, little brother. So SMN1 is sort of the big brother that's really good at making this protein, and SMN2 is the little brother that tries but can't quite keep up. So you can see that SMN2 in wild type cells or normal cells that are not treated usually only produces a functional SMN protein about 10% of the time. About 90% of the time, SMN2 is creating this non-functional truncated version of the protein. And the difference here is that sometimes the gene is incorporating exon 7. So this is sort of the difference between SMN1 and SMN2 is this exon 7. And then when it doesn't incorporate exon 7, it's not creating a functional copy of that protein. So the way that Spinraza works is it actually encourages the body to incorporate that exon that's being skipped and making SMN2 less efficient. So by adding the exon back in, that SMN2 gene is able to produce a full SMN protein. So that's kind of compensating for the abnormal SMN1 gene. So in this case, we're kind of using that ASO to alter what the gene is naturally doing and making it do it a little bit better. In Melicin, there's sort of a different mechanism. So the ASO is being used to correct a specific genetic mutation that's unique to one patient. So in a normal CLN7 gene, the body is going along from exon 1, 2 through 5, exon 6, and then exon 7 and so on. So it's incorporating all of these exons the way it's supposed to. The mutation that Mila has introduces this region of DNA that kind of throws off how the body is reading those exons and causes it to stop short. So the rest of the protein is not being made, and it's resulting in this version of the protein that gets broken down and degrades, and so isn't having a function in the body. So the way that Mealison works is it encourages the body to incorporate these exons the way that we want them to be incorporated. So it's going from exon 6 to exon 7 in a higher proportion of cases than it does without the treatment. So you can see when these cells are treated with Mealison, about 32% of the time, the body is doing, it's making the correct version of the protein and skipping over this abnormal region. So basically, this isn't perfect. It's not restoring full function, but it is allowing the body to create the correct protein a larger percentage of the time. So that's going to give us, or give Mila, higher levels of functional protein. So basically the hope is that ASO therapy might be able to work for some CDKL5 patients. We think that maybe 15%, maybe a little bit less, have splice site mutations. Not all of them are going to be perfect for ASO therapy. Kind of just like the nonsense mutations, not every nonsense mutation is a perfect target for atalurin, and not every splice site mutation is going to be a perfect target for an ASO. But I think there are definitely two potential exciting areas. We've got a long way to go, but we're making good strides. So here's some of the resources that I'm sure most of you are very aware of, references. And I just had to prove after all the cat analogies that I'm not a crazy cat lady. So this is a picture of my dog, but I do also have a cat. Before I wrap up, I just want to thank IFCR for hosting and making this happen virtually. I know that was not an easy feat. And thanks everyone for joining us. Dr. Binky is going to be joining us pretty soon, so just stick around for his talk, and then we'll both be around for questions afterwards.
I might ask you a question since I have you here. You discussed splice site variants and opportunities for therapy there, and then also Xinactivation in girls. I found the skewing of the Xinactivation interesting. As caregivers of females start looking at this, how is the skewing detected, and how might you learn whether treatment is available to your child, given whether or not they have that as a characteristic of their mutation? And especially because you mentioned that it relates to severity and clinical symptoms and outcomes.
Yeah, so there is a separate genetic test that can be done to specifically look for Xinactivation. It's not going to tell you the proportion of cells that have the mutation and the proportion that don't, but it can tell you percentage of how skewed that Xinactivation is. So typically the proportion of one copy to the other will be somewhere around 50%. Usually it's a pretty wide range between 40 and 60. But with people with skewed Xinactivation, it's going to be more like 80, 90, maybe higher. So there is a test that can tell you that percentage. Obviously, it's only going to tell you the percentage within the sample that's tested. So the blood or saliva or whatever is looked at. So we can't perfectly apply that to the brain, but we can it's at least a starting point to understand if that's something that's happening.
So the tissue type is important, where you're looking, but you mentioned timing several times. So the age of the child being tested, is that also important on whether you might see this or not?
Not really. So the Xinactivation is something that's going to happen. It's going to be most important early in development. So once that happens, you know, we always have new cells that are forming, but in our brain, things are pretty much staying the same. So once one of those copies is turned on or turned off, it pretty much stays the same throughout that person's life. So there is, you know, higher cell turnover in different types of tissue, skin, blood, those sorts of things. But usually the level that's present in the blood is not something that changes greatly over time. If it's skewed, we're usually going to see that it's skewed early on or later in life. So we might test a patient if we suspect that. We might test a parent if we suspect that. And in both cases, we can detect if it's skewed.
And you may have answered this, but does that have any implication on the ability to reactivate the X chromosome? Yeah, the normal. Mm-hmm.
I'm not really sure.
Either.
I think you might kind of be at a better starting point if more of the, you know, if more of the normal copies of the cell are turned on to begin with. You've got sort of have less work to do. But yeah, I'm not sure if that's been looked at yet. I think we're still at the beginning stages of exploring that as a therapy.
Thank you.
Yeah.
So we are expecting Dr. Binky soon. But in the meantime, we could just take a break, I suppose. Karen, we can thank our sponsors again.
For sure.
And let Katie have a little break before she has to come back and join Tim.
Amanda, if you're listening, do you want to share those sponsor slides again?
Yep, they're coming up.
Awesome. Thank you.
Thanks, Katie.
Thank you so much, Katie. And I will mention, I know we had shared Katie's talk from Denver in 2018, but if you're tuning into this and you didn't know that there was a more basic genetics talk on our website, please check that out. She did a beautiful job then as well. So if this was a little deeper and harder for you to understand, it might be helpful to go re-listen to the talk she gave in 2018 that we shared a few weeks ago.
I'll also mention Genetics Home Reference is a really great resource for anyone who does want to kind of explore more of those basic genetic concepts. It goes over all sorts of different things that we touched on in the last talk and really didn't talk about much today. So that's somewhere that I would point people if they want, you know,
Katie, is that a website or a book? Or...
Yeah, so Genetics Home Reference. So it's, I can put it in the chat if that's helpful. So it's through the NIH. So it is ghr.nlm.nih.gov. So if you go to that website, you can look at CDKL5 specifically. You can go to, there's a link on the top on that main page that says, help me understand genetics. There's videos, there's all sorts of different links that you can click on that will explain different topics. So it's a really good place to start.
Okay, thank you. And then we can share that link on the Facebook post and also on the chat for the YouTube live so that it lives there and people can find it.
Katie had a nice, the slide from the Khan Academy has some nice schematics of how some of the different treatments work and splice variants versus not, you know, it walks you through it in a way that's easy to understand.
I'll chime in that they have a lot of videos even that dive into very complex genetic scenarios that make it very easy to digest too at Khan.
Khan Academy is a great resource.
For sure.
As well. So do we want to, I mean, if people want to take a break, grab a snack, rejoin, I will mention, I think Tim was, when our conference moved from in-person to virtual, people institutions wanted them to see patients today. Go figure, they didn't want them off work. So I think Tim is actually seeing a patient and it might have run a little over. And so just bear with us if he's which he's not late yet, but just bear with us if it is a few minutes, and just keep listening and you'll know when he joins. This was, Amanda and I feel like this was much more difficult to organize than an in-person conference. It's been a learning experience, and I think we're learning as we go.
I think we're all learning as we go when it comes to Zoom and making things work in the age of COVID.
Absolutely, absolutely.
I'm glad you guys were able to put this together. I'm definitely missing seeing everyone in person though.
Right? I would have loved to have seen you again, Katie. It's very sad. I have actually, I think this is the biggest loss I feel like I've suffered during this, because to me, in-person conference is just so inspirational and rejuvenating and helps everyone plug in. And I hope that we can capture some of that with this effort that we're doing.
So I have a question, Katie, that's come in,
Yay.
and I'm not, I will read it to you. And Bob, if you're listening, go ahead and chat back with me if you need to go into more detail. Bob, they got their diagnosis yesterday and they have a 4.5-month-old daughter. He is wondering if there's any way Xinactivation could be impacted at this point.
So it sounds like that's very young. So it's hard for us to tell that early on exactly what the severity looks like. I would say that it's definitely something that could be tested. If you're interested in Xinactivation testing, I would bring that up with your child's doctor. That is something that they could send. I don't know how useful it will be this early on, but it might be something that's good to know about. I would also say, you know, it doesn't have to be something that you are thinking about right this moment. I think right now with a brand new diagnosis, the focus is just figuring out what to do with that information, figuring out how to take care of your child. But one of the things I would sort of keep in mind, maybe a little bit further down the road, is thinking about potentially doing parental testing to see if that mutation might be present and if there might be a chance of it happening again in future children, because that's another place where Xinactivation can come into play. Is if mom has a mutation and has skewed Xinactivation, she might have a mutation like this and not know that she has it because she's not actually expressing any of those symptoms.
Thank you. And did y'all say his name was Bob? I can't see the question, but I think you said Bob. So I would just say, be patient and kind to yourself. And there will be a lot of information shared over the next couple of days, but just remember, it's all going to be videoed. It's going to live on the website, so you can review it as needed, or even take a break and step away from it and come back and watch it later as well.
And if you don't have a genetic counselor that you've worked with, or you don't have the opportunity to meet with a genetic counselor, if you have any specific questions about your child, feel free to reach out to me. I'm happy to, you know, to help walk through the report or answer any of the questions that you might have. And Amanda, you can share my email. I put it at the end of the slides. But
okay. And so, Katie, we have some more questions coming in as well. So
okay.
while I'm going to try to keep my eye out for Tim to pop in here. And in the meantime, though, we have, okay. One, somebody said, thank you. It's very informative. Two, somebody that's catching bits and pieces in between work, but looking forward to watching later. Okay, so those are more comments, but let me give you, I don't know, what kind of timeline are we looking at for gene therapy clinical trials in humans? And what are the risk factors besides from introducing new changes to the gene.
You
So
make those big decisions, don't you, Katie?
Yeah, that's a good question for Tim.
I
Tim
think
put
that
one
Tim's
on
going to
me.
cover that maybe.
Yeah, I think it's always hard to answer those questions, especially with how chaotic everything has been over the past several months. A lot of timelines have been pushed back. We've had to you know, hold off on research visits in some cases. So I think that question is probably harder to answer right now than it ever has been. But yeah, ask Tim.
He's connecting
Yes,
right
come-
now.
To him.
Yes, I'm going to mute out. So
Hello, Dr. Binky.
Hello. Sorry to be late, to keep you waiting.
It's okay. We found stuff to talk about.
Okay,
It's all good. Thank you for joining.
so I'll share my screen now.
Do you have permission to do it? I think you should.
I think so.
Okay.
Great. Can you see my screen now?
Yes.
Yep.
Great. All right, will you tell me when I should start?
We're ready for you whenever you are ready. Let me first introduce Dr. Tim Binky, director of our CDKL5 Center of Excellence and MD. He has all these credits to his name that you can see on the slide. I'll let him take it from here. He is the medical director at the Rett Clinic, though, and the research director at the Neuroscience Institute of Children's Hospital, Colorado. He's going to talk to us about the hot topics, giving us some research updates and updates on COVID-19.
Great. Well, thanks, Heidi, Amanda, Karen, everybody, for asking me to do this. It's fun to be here. It's a little weird. I miss seeing you all out there. But here, let me see if I can make this thing go forward. Okay, so here are my disclosures. And I think that we're recording this. If there's any questions or concerns about any of these, then please let me know. So here's what we're going to talk about today. So we're going to have a few thoughts about COVID-19. Then I would like to go through some older stuff in the literature, basically to provide some historical perspectives. We're going to talk about some brain basics, and then we will talk about cyclin-dependent kinase like 5, the gene and the protein. Then I will go over some newer stuff. I want to talk about how to evaluate the literature. I'll talk about the new things that have come out in the last year, including one of the gene therapy trials in a rodent model. And then I'm going to have a few comments about some off-label uses that I think are reflective of some of the recent research. And then we'll go through some conclusions and then get to the question and answers with Katie and myself. So, COVID-19 updates. So I've been posting as medical advisor for rettsyndrome.org to the rettsyndrome.org website. And these other comments have also shown up on the Rocky Mountain Rett Association website and the IFCR website, and I've had a lot of good discussions with many of you in the community that have informed these thoughts. So the first thing I want to say is that we have not had any official reports. In other words, there's nothing been reported in the literature or anything formal like that of COVID-19 infections in our patients with developmental encephalopathies. So Rett, MECP2 duplication syndrome, CDD, FOXG1, et cetera, in that community. And while we continue to remain vigilant and concerned, so far, those few anecdotal reports, and there are a few anecdotal reports that we're aware of, did not include the need for ICU care. But if any of you know anything differently, I would welcome you reaching out so that we can stay informed in that regard. The current goal that I think that we need to think about as a community is the safe return to schools and therapies. And in order for that to happen, we all, that's the whole community, need to practice frequent hand washing and mask wearing to protect all of us. And the people that I'm thinking about are not just our patients, but our school teachers, our therapists in the school, and everybody around them who potentially could get sick with this. And so mask wearing and hand washing. Another aspect of safe return to school requires flu vaccinations and COVID vaccinations for all teachers, children, and family when these are available. Another important thing to mention, and that's why it's underlined here, is remember, if your child needs medical attention, get it. Don't not go to the doctor if your child needs help. At Children's Hospital here in Colorado, it's designed to be as safe as possible. Everybody working at Children's, we're screened. We have access to rapid testing. If people are staying in the hospital overnight, then they get rapid testing. We're all wearing masks and face shields. So we're making it as safe as we can possibly get it. The other thing to say is stay safe out there. We miss you and we really look forward to seeing you soon. Okay, let's jump in and go through I know there's a lot of old hands here, but there's also probably a lot of new hands as well. So let's talk about CDKL5 deficiency disorder. It might be boring for some, but I think it's useful to go back to the beginning just to make sure that everybody's on the same page. CDKL5 deficiency disorder, or CDD, it's a developmental encephalopathy. That's the fancy word meaning for the symptoms change with age. In 2004, there was a paper that described it as a genetic cause of medically refractory epilepsy beginning in the first year of life, with intellectual disability and global impairments. It was for a while called a Rett syndrome variant, but we know from the work that's come from Helen Leonard and Jenny Downs that this is really its own disease. But prior to this, it's been a lot of combined case reports, clinical series, and surveys. But now through the Centers of Excellence, we have a picture of a larger clinical cohort, which we've published last year. So cyclin-dependent kinase and cyclin-dependent kinase-like kinases. It's a mouthful, but what's in a name? So cyclin-dependent kinases regulate mitosis. This is the process of how cells divide. And there's been a lot of interest in these because these cyclin-dependent kinases, they're involved in cancer. And the diagram here talks about showing the cell cycle, which is something that gets stuck in the reproductive phase in cancer. And this fancy diagram is just showing how there's a lot of different cyclin-dependent kinases that are involved in mitosis and also cancer. As a result of that, there's a lot of interest in the pharmaceutical industry in cyclin-dependent kinases. They regulate growth and functions of something called the primary cilia. So as a result, CDKs are involved in how cells stretch out, move, and grow. And this is a part of normal growth, but it's also potentially a part of cancer as well. Cyclin-dependent kinases bind and are regulated by a protein called cyclins. Cyclin-dependent kinase-like kinases, they have cyclin binding domains, but they don't bind cyclins. They bind other things other than cyclins. And all of these things are kinases. That means that these proteins turn other proteins off or on by adding a phosphate group to them. And here's the diagram. So CDKs, they bind cyclin, that activates them, allows them to put a phosphate on some particular protein, and then this particular protein does whatever it's supposed to do. So what are the molecular genetics? And Katie talked a lot about this, I think, in her talk, but the molecular genetics have told us that missense mutations, we've learned that the catalytic domain is the primary functional domain of cyclin-dependent kinase like 5. And here is just when you take all of the little chunks of RNA and you put them all together, these are the instructions for how to make the cyclin-dependent kinase-like protein. And these are some of the different mutations that we've learned about that can be missense. But there's also, of course, as Katie told you about, there are nonsense mutations as well. We've also over the years found some that don't seem to affect function. And I'll talk to you about more of those in a minute. But through genetic studies, we know that the incidence is approximately 1 in 40,000 births. And we know some information about prevalence. So from the Centers of Excellence, we follow about 175. Amanda, I think, that has told me this is a number through the Facebook estimate. We know from Colorado, we can guess that it's about 1 in 75,000 based on the patients that we've followed, because most of the patients here in Colorado tend to come see us because we're the one center. But this chunk of RNA, it gets turned into this beautiful protein, and basically what all of these little twists and turns, they're describing the three-dimensional structure of the protein. And you can almost see there's this little mouth-like thing here, and you can imagine that this is the mouth where proteins get sucked into the kinase, and that's where it puts on the phosphate group. And the little red dots are where there are disease-causing mutations, but there's also a lot of blue dots out here where there are changes that aren't affected. So in other words, these are changes that we see in otherwise normal individuals. And those show up in this fancy database that we use to track those called Nomad. So I want to talk to everybody briefly about brain basics and talk about synaptic transmission, because this is really important when we think about what CDKL5 is doing. So in the brain, you have excitatory synapses and you have inhibitory synapses. But basically at excitatory synapse, there's a wave of depolarization that we call an action potential. And I can't do it so well because my arm's in a cast, but it's a wave of depolarization that goes along this axon, and when it hits the end or the terminal, it causes the release of glutamate. Glutamate diffuses across a space called the synapse, and it binds with receptor channels on the other side, and those receptor channels allow the influx primarily of sodium. Sodium tends to depolarize the neuron on the other side. Now, in the case of an inhibitory synapse, we have that same traveling wave of depolarization. It runs into the end at the synapse, and in this case, at an inhibitory synapse, it causes the release of GABA. This neurotransmitter diffuses across this space, binds to receptor channels, and in this case, allows the influx of chloride. Chloride actually causes a hyperpolarization, which we call it's inhibitory. So glutamate receptors, there's a lot of different types. There's an AMPA type, there's an NMDA type. For GABA receptors, there's a lot of different types. But basically, what synapses are doing, there's this bridge between electrical communication from one neuron to the next. So synapses are this chemical bridge for electrical communication from one neuron to the next. And what's important on the neuron on the other side is as if this sum total of depolarization and hyperpolarization is actually enough to reach an excitation threshold, then that electrical signal will keep moving through the system. And what does that system look like? So here's my Brain Basics 2 slide. Synapses are part of a communication network. So if we have one neuron here and there's an axon, and let's say that down this axon there's that traveling wave of depolarization, there can be a branch that goes back to that and touches back onto itself, which we call positive feedback, or it can go on in the circuit to either make a synapse onto an inhibitory neuron, or it can make a synapse onto the next neuron in the chain. And if the sum total of the excitation and inhibition is enough to reach a threshold, then the communication continues to move through the network to the next neuron, and so on and so forth. Well, this is a useful way for information flow, but if you have this sort of positive feedback, that's actually a good way for having memory storage, or as you can imagine, it might also be an accident waiting to happen. which can be a seizure as well. This sort of positive feedback is really important in a number of circuits, such as the hippocampus, which is where learning and memory takes place, or the thalamus, where a lot of gatekeeping of moving information in and out of the brain happens. Brain Basics 3. Well, there's even an even smaller communication network of synaptic proteins. So here's an example of one of those synapses here. And they make contact for excitatory synapses on these little protrusions called dendritic spines. Well, if you blow one of those up, there's a whole lot of stuff inside both the front side, the presynaptic side, or the postsynaptic side of this communication happening at this synapse. And here we have drawn out for us now NMDA receptors and AMPA receptors, and then all of this stuff inside, which is holding them all in place. And this stuff that's holding them all in place is a secondary communication network of synaptic proteins. And one of those important things that lives there are kinases. And one of our favorite kinases, CDKL5, it lives in this space right here. So what did we know about the physiology of CDKL5 and the pathophysiology of when it's not working? We know that CDKL5, it's localized to both the nucleus of cells, but also to the dendritic spines of excitatory glutamatergic synapses. So glutamatergic means you've got those AMPA receptors and NMDA receptors there. Well, CDKL5, it's thought to be a kinase and phosphorylates MECP2 and something else called DNMT1. At the synapse, we think that it's phosphorylating something called EB2, NGL1, amphiphysin, and we're still learning more about the other things that it's phosphorylating. In other words, these are things that CDKL5 is turning off and turning on to do the things that it's supposed to do in the synapse. While it's there, it's stabilizing the formation of synapses, and it interacts with this very important protein called PSD95, which is the glue that seems to hold a number of receptors in the synapse, also colibistin, ISIN-1, and GL1 actin. And we know that if it's not there, you tend to have too many of a certain flavor of NMDA type glutamate receptors called gluin2B. And through studying abnormalities in rodent models and culture systems, we know that CDKL5 is really important in behavior, how excitatory synapses work, visual function, and it's also started to suggest some therapeutic approaches. And here are some of the literature, and we've gone over some of these in past literature updates. So when you think about evaluating, oops, something happened. Let me try to get it going again. Can you guys hear me still okay?
Yeah, I can hear you.
I can
Okay.
hear you.
All right, I don't know what happened,
There
but,
it is.
but
It's up there.
my computer just decided to stop and make me start at the beginning. Okay. Almost, oh, here we are. Okay, we're back to evaluating the literature. So the way I've gone through some of the interesting papers over the last year, I tried to, the eye that I take to it is one that I think is, I want to put myself in, what if I was trying to figure this out for my own child? So does this paper tell me anything about my child? Does it tell me something about prognosis, symptoms, etc.? Does this paper tell me anything about possible therapies for my child? And so when that's a possibility in the paper, you ask, well, was it a human study, which might be better than a study of rodent models, which might be better than a study of cell culture models of CDKL5 deficiency disorder. And I put quotes around better than because they're not really better than, they're all providing information, but it might be a little closer to where you're thinking of how close are we for taking this therapy into the clinic. And the next question is does this study map onto how I could envision my child being treated? And so you're asking the question, does the timing of the treatment in the study map into reality. In other words, we know when we're diagnosed, we know when we get our tests back, we know where we are now with our children. Is that therapy relevant to where I am now with my child? An important question then is to ask is, did they use the correct control groups for the right comparisons? Are they asking the right questions to ensure that what the therapy is doing is really improving things in whatever model they were using? And most importantly is, did they use that to address safety? Is what they're going to do really going to help things, or we don't want to throw the baby out with the bathwater here when we try to approach therapies? The next thing to think about is, my analogy is, how many swallows make a summer? Well, here's a swallow up here, and this is just one little swallow, and you can't tell, but it's actually winter in the background. So we're not summer yet. Here we've got 3 swallows. Still probably not summer yet because you can't tell, but it's not really very green back here. Oh, we've got now a ton of swallows here. Must be summer. I'm using this analogy because reproducibility is a serious issue in science. You've got to have a lot of swallows before you really think that it's gone from something that's really a concept that's got some proof behind it. So other labs must replicate the results for translational studies to become proof of concept. So the flow is a lot of translational studies prove a concept, then you move into a preclinical realm where you set up things exactly as you would do in the human, but do it in your rodent model, prove that it's going to still be safe, and then it's time to move towards clinical trials and approval. And this is what the FDA looks at, is, have they done their homework to say that it's safe first before it's ready to go to clinical trials, before we can get anywhere close to approval? Okay, with this sort of backdrop, that's how I went through some of the papers over the last year. So I think Eric's going to talk about this paper more tomorrow, but it is a paper that came over the last year, just in case he didn't get to it or didn't want to, because it was really a big effort on his part to do this study. But the title of the paper is Comparison of Core Features in 4 Developmental Encephalopathies Seen in the Rett Natural History Study. It was by Coutry French and Eric Marsh and the rest of us in the Rett and Rett-related disorders natural history study. So in this study, there were 535 patients that had typical Rett syndrome and 53 patients that had CDKL5 deficiency disorder. And if you just look at this graph up here, in typical Rett syndrome, these patients have smaller head sizes. And the way that we're plotting it out here, this was a lot of work on Eric's part to come up with this plot. But basically what this is showing here is that on average, Rett patients have head sizes that are 2 standard deviations below the mean. And that's what this graph is showing. This is showing the broad range that's seen there. Whereas patients with CDKL5 disorder, they tend to, it's just 1 standard deviation below the mean. So still on a growth chart, but there are some patients that have smaller head sizes. So one of the questions was, does this mean anything? Well, at least in terms of Rett syndrome, if you plot out the severity score versus the head circumference z-score, there is a suggestion that, not a suggestion, it's the statistics bore this out, is that more severe patients have smaller head sizes in Rett syndrome. And this is using the Rett syndrome specific clinical severity score. Now what you see is there's a lot of dots around the line here. Now if all the dots were exactly on the line, then you'd have what we call an R squared of 1. But you've got dots that are away from the line, and so the R squared is less than 1. So in this case, the R squared was about 0.3 for Rett syndrome. When you do this sort of plot for CDD, the R squared was smaller. It was about 0.2, but it was still following this sort of line. But the other issue is that the severity score that we used was actually a Rett specific score. And so it will be interesting as we move forward with a lot of the other studies in the ICCRN to see with more specific scales how this sort of thing is really borne out for CDD. So it's a great paper, a lot of work on Eric's part. Okay, and more of the translational science. So one of the papers was this one: fingolimide modulates dendritic architecture in a BDNF dependent manner, by Potnick et al. In the International Journal of Molecular Science. So what they did is they treated cultured neurons from rodents that were completely lacking the CDKL5 gene. They then treated these cultures at 7 days in vitro. That's what DIV stands for. So in other words, these are very immature neurons in a culture dish. And they found that treatment with fingolimide improved dendritic complexity. So this is what I would call a translational study. It suggests that fingolimide might be interesting to understand how it works, to try to look at this in a lot further detail. Here was another study. It's titled "The Green Tea Polyphenol Epigallocaltechnin 3 Gallate," or easier to say, EGCG, restores CDKL5 dependent synaptic defects in vitro and in vivo, by Trovo et al. In Neurobiology Disease. So they treated cultured neurons from rodents and mature rodents. And the mature rodents, when they treated them with EGCG, they treated them for 30 days. And in both situations, it improved the number of dendritic spines. And you remember what dendritic spines are from our picture earlier. But there was no improvement in learning and memory behaviors in these animals. So one of the questions is well, they treated mature rodents. What if they treated rodents when they were much longer? Would they still have, what would we have seen? Would it have been any different? The other thing to point out is they did something that was real world. It was a chronic treatment, which is you'd want to know, if I'm treating my child every day with something, what's the end result? That's what's something that's very relevant. However, they only looked at a single dose. So they didn't try a number of different doses, and they didn't do any pharmacokinetics. So we're not really sure how much of the EGCG really got into the brain. So what I would say is this is a translational study, and it's what I would call a single swallow. It's important, but it's a single swallow. There was another study titled "Pharmacotherapy with Sertraline Rescues Brain Development and Behavior in a Mouse Model of CDKL5 Deficiency Disorder." It was by Fuchs et al. In Neuropharmacology. So they looked at adult female knockout mice, which is relevant for what we're all thinking about. So these, most of our patients are females, but it's one of the appropriate models that you should consider. Also, they treated as adult. They treated for 30 days. They found that it improved dendritic structure. It improved learning and memory, which is really exciting. But they used what is relatively a high dose for sertraline, and there were no pharmacokinetics, so we don't really know how much got into the brain. So this is an interesting study, but it's what I would consider translational, and it's a single swallow. Another study, pregnenolone and pregnenolone methyl ether rescue neuronal defects caused by dysfunctional CLIP170 in a neuronal model of CDKL5 deficiency disorder. So this was by Barbiero et al. It's in Neuropharmacology. So they treated cultured immature neurons from rodents completely lacking CDKL5. And what it did is it investigated the specific CDKL5 signaling pathway associated with microtubules. So that's all that stuff that's sitting in dendritic spines where CDKL5 is moving all of that stuff to sit into the synapse. So this is a very interesting study, helps us understand more about how CDKL5 is interacting with microtubules. So it was a translational study. The next study was AMPA receptor dysregulation and therapeutic interventions in a mouse model of CDKL5 deficiency disorder by Yenawar et al. In Journal of Neuroscience. They looked at male mice that had a human mutation knock-in of CDKL5. And they found that one flavor of AMPA-type glutamate receptors were GLUA2 lacking in these adolescent and adult mice, whereas it seemed to be normal in the immature and younger adult mice. They found that there was abnormal behavior in these knock-in mice, and when treated with a single dose of a drug called IEM1460, which blocks these GluA2 AMP receptors, with a single dose, so single dose, test them a couple hours later, their behavior was corrected by this. They also found, interesting, that the seizure threshold was corrected in a similar fashion by the single dose of IEM1460. It's a very interesting study, translational in nature to help us understand about what CDKL5 is doing at synapses, but it's a single swallow. And it doesn't really directly translate because it's this single dose of a drug that wouldn't really be applicable to a real world situation. The next study was altered NMDA receptor signaling underlies autistic-like features in a mouse model of CDKL5 deficiency disorder. This was by Tang et al. In Nature Communications. So they looked at adult male mice with a knock-in of one of the human mutations. It was one of those truncation nonsense mutations. So again, because these are male mice, these are completely lacking in CDKL5. They found in this model that there was increased expression of one of those flavors of the NMDA type glutamate receptors of GluN2B. And they found that a single dose of memantine, which is an NMDA receptor antagonist, improved social behavior, but not learning and memory in this model. They did try multiple doses, and the doses that they picked were based on pharmacokinetics. So they feel that using these doses, you are getting something that's relevant amount into the brain of the mice. So this study, it's translational in a lot of aspects with a number of the different studies they did, and I'm calling it a second swallow because this is additional evidence that in another model there's increased GluN2B flavor of NMDA receptors. So very interesting and helpful study. Here's the next paper. Splicing mutations impairing CDKL5 expression and activity can be efficiently rescued by U1 siRNA-based therapy by Balestra et al. In International Journal of Molecular Science. So they modeled a human mutation that affects splicing. And they looked at this RNA-based therapy and found that it rescued a dendritic phenotype in immature cultured neurons. And the RNA was delivered by lentivirus transfection. And the question is this type of RNA-based therapy potentially amenable to an antisense oligonucleotide or similar type delivery method? So it's not clear if that's the case, but this could be something that's interesting for the future. So very interesting study for some of our patients that are affected by splice site mutations. So it's translational in that regard. The next paper was artificial escape from X chromosome inactivation, or XCI, by DNA methylation editing of the CDKL5 gene by Helme et al. In Nucleic Acid Research. So they looked at non-neuronal cells in culture, and they found that with some a clever approach that it's possible to turn on the other copy of CDKL5. So this is really exciting, but there's clearly a lot of future work to try to translate this, but it's a breakthrough because you need to move from, what do you have to do? You got to move from non-neuronal cells and cells in culture and figure out how to deliver in that type of system the sort of machinery that they delivered in a dish in order to make this sort of thing happen. So translational. The next study that I think that we want to talk about is CDKL5 promotes proliferation, migration, and chemotherapeutic drug resistance of glioma cells via activation of the PI3 kinase AKT signaling pathway by Zhang et al. In Feb's Open Biology. So what they found is that overexpression of CDKL5 may drive the formation of gliomas. So gliomas are a type of brain tumor. And the reason why I thought this paper was interesting is because it raises safety considerations for gene replacement therapy. The next paper to talk about is a kinome wide screen identifies CDKL5, SOX9, regulatory axis, and epithelial cell death, and kidney injury. So what this was by Kim et al. In Nature Communications. So what they found in kidneys is that SOX9 is one of those proteins that CDKL5 phosphorylates. And that activity mediates acute kidney injury. So acute kidney injury is something that happens when you're severely dehydrated, you've got poor blood flow to your kidneys, and they get injured. And what they found in this study, interestingly, is if you have genetic loss of CDKL5 or its blockade, this is protective of acute kidney injury. So again, I think this is interesting, but it also represents a safety consideration for gene replacement therapy. In other words, you don't want your kidneys to have too much CDKL5. It might make them vulnerable to acute kidney injury. So it's just something to think about, keep in the backs of our minds. So the big paper to talk about is this one. Gene replacement ameliorates deficits in mouse and human models of cyclin-dependent kinase-like 5 disorders by Gao et al. In Brain. So they used gene replacement to adolescent male CDKL5 knockout mice, and they were then investigated 30 days later. So again, these are male mice, so they're completely lacking in CDKL5. They were at an adolescent stage of development, and they were then investigated 30 days later. They used gene replacement with a mouse-specific adeno-associated virus, that's AAV vector, and it was given intravenously. The reason why I say it's mouse-specific is this particular vector really likes to infect the brain cells in mice. They found that gene replacement was brain region specific and variable. So in other words, they found that there was gene replacement in the cerebellum of up to 60%, but in the cortex and in other areas, it was only 10%. They found that in the mice that this improved coordination, but not necessarily learning and memory. And that probably goes along with, if you have more replacement in cerebellum of 60%, you're going to improve coordination. But it didn't replace things in cortex, so you might not have expected necessarily that it would improve learning and memory. So this is an exciting initial translational study. I was asked, along with Dr. Peter Kine in Edinburgh, to write a commentary of this that appeared also in Brain. So some of the things that we thought about when we looked at this paper is when they developed their CDKL5 knockout mice, they actually had an unusual breeding strategy. And this is a nitpicky point. The other thing is when we're talking about the mouse-specific AAV vector that they used, this vector, it turns out, may not be safe in non-human primates. So this is an important study in terms of proof of concept, but it does not necessarily say in any way, for a lot of the reasons that I'm going to talk about, ready to move on to the next step in terms of preclinical studies or even human studies. One of the aspects is it would have been nice if they'd done a lot more controls and treatment groups with the way that they set up the study. And what I'm talking about is we would like to know, is there an effect of this replacement, not only in these male knockout mice, but what about female knockout mice that are heterozygous, or even wild type mice, to prove that this sort of therapy is not going to cause any harm. Those sorts of controls are absolutely essential. One of the things that they did is they used an intravenous route. Now that was an appropriate route for this particular vector that they used to get the sort of expression that they wanted to see. But this route may not be suitable in humans because the vector is felt throughout the body and it generates a large immune response, which is in some ways like shooting yourself in the foot, because that immune response is going to get rid of the virus that you really want to do the job for you. The readouts of gene replacement were indirect. And so it left asking the question, is the replacement CDKL5 kinase, how much of that they put back in, is it functional? And when you do gene replacement, the business end is what we call the promoter. It's this little bit of DNA that's in front of the CDKL5 gene that tells you how much to make of the gene. And the promoter that they used was prolific. In other words, it's not regulated. So we don't know whether or not there's too much CDKL5 in each cell. And as we talked about in some of the other studies, this is an important question. We know in some humans that there could be something called CDKL5 duplication, which is the cause of developmental concerns in these patients. And so it's something that we need to ask and address. Is too much CDKL5 an issue? There was also some, it's a first-time study. A lot of these things can't be addressed, but what you really want to know is something about long-term safety. Are other brain regions, such as the dorsal root ganglion, organs, liver and kidney, are these okay with this type of treatment? But that wasn't examined. Another thing that we all want to know about is what about seizure thresholds? And this was not examined. So this is a very exciting study. It gives us a lot of hope that this sort of approach is something that will be helpful, but it's one swallow. So all of these things bring about the question of, well, okay, doc, you're telling me it's one swallow, but what about my kid here? Should my child be treated off-label with any of these things that are suggested with green tea, with sertraline, IEM 1460, memantine, fenfluramine, etc.? So as a parent, what should you be asking? If these things are brought up, everybody needs to be asking, is it safe? And does the clinician involved have experience with this medication? And so the question is swallows. Has this medication really been proven to be helpful? And we have to ask, even in if there's small clinical trials, there is the potential for placebo effect, even in terms of counting seizures. The next question is even if you're prescribing it off-label, it doesn't mean your insurance will cover this. So will my insurance cover this? Insurance is unlikely to cover things like memantine and certainly fenfluramine, unless you have a specific diagnosis. And so I think another question is when can I enroll my child in a clinical trial for this? And I put some things here as, I don't think that we ever want to be seen as pushing snake oil, and we certainly don't want to be barking up the wrong trees with anything that we're thinking about when it comes to patients and patient safety. So we've gone over a review of CDKL5 and brain basics. We've had a review of what to look for in the literature, review of some exciting progress in the field, and we talked about the importance of being safe out there. So I want to thank you guys, our patients and families with CDD. I want to thank everybody in the Rett Clinic, Scott, Tristan, Katie, everybody else, and all of our collaborators within the ICCRN, Helen and Jenny, NIH Natural History, Eric, Alan, Jeff, Ralph at University of Glasgow, Scylla at Francis Crick, and Peter Kine at Edinburgh. Certainly Rocky Mountain Rett, who supports our clinic, IFCR, who supports our clinic, Lulu Foundation, who supported our research in the past, and we also enjoy working with them, and through the Ponzio Family Chair, which supports me in neuroscience research. So I think that brings us to the end of my talk. So I can quit sharing, if I can figure out how to do that, stop share, and we can go to questions.
Thanks,
Hi,
Tim.
Tim.
Amanda, do you
Hi.
have any questions?
I do actually. Do you, Heidi?
Not that have come in through the email box
Okay.
now.
So the first question I have is, are we going to, oops, sorry, let me, will boys be left out when it is time for initial gene therapy clinical trials?
So first of all, I will say that is not up to me. This is a decision that when a drug company is developing this, that they will make those decisions. those decisions are based on how can we get the most subjects into a trial. So that's one of those factors that's around male patients. And the next is, how can we be sure that when we assess all of these patients, that are we going to be comparing apples and oranges too much? As part of, I hope, one of the people that's involved where they might ask, hey, what do you think about this? I would suggest that I don't see any reason why not including males in trials, because if they meet criteria, they meet criteria, whatever your criteria is for getting into the trial. So I would certainly hope not, would be my answer to that. But I don't know the answer until we finally get there. Do you think that that answered their question, and would you want to follow on to that, Amanda?
I believe that answered their question. If not, go ahead and message me.
Okay.
I'm going to follow up with another question, Tim, and this has to do with cholesterol. And this mom says, I understand there is a link with high cholesterol and Rett syndrome, but is it unexplored in CDKL5? She has in infants and older children. She had an experience where her 10-month-old had very high cholesterol levels in her pre-keto workup. And she says that multiple parents have also reported this when she chatted about it.
I think that might be a question for our epileptologists to say what their experience is and how often they find that as part of their keto workups. In terms of high cholesterol is a relative, and predisposition to this is a relatively common thing in the general population. And is this something that's seen more in the ketogenic diet population in CDKL5 patients, or is it just is it really seen more often in CDKL5 patients? And the answer is we don't know. I mean, one of the studies that was referenced does have to do with modifying cholesterol synthesis, and that's the fingolimide study, which again was very translational. So I think that it's something that's definitely worth exploring. But I'll defer other answers with regards to whether or not folks might think this is something there who are seeing a lot of patients in the keto clinic.
And we could ask that question maybe when we do the optimizing epilepsy treatments panel of the clinicians at that time. We'll keep it for tomorrow.
Okay, so I know with the live feed, sometimes there's a bit of a lag. I don't see any other questions. We have a lot of comments that have came in. I think for the community, if you would let us know if we're overlooking any questions you have, be happy to ask. And if not, we'll certainly help you follow up with getting answers to any of the questions you have after watching this, if you're not here live, from Tim and any other folks that might be able to help us answer what you're looking for.
Well, Tim was in transit. There was a question for Katie regarding how far away we may be from ASO and gene therapy and whether there might be age limits on these treatments. She passed it off on you.
Thanks a lot, Katie. So I think that we're hopeful that there will be a gene therapy trial sooner than later, but in terms of the time frame, I'm not directly involved in any of those, and if I was, I couldn't tell you because somebody might have sworn me to secrecy because of those sorts of confidential agreements that exist. But I think one of the things that I want you to take home from my discussion of that one paper that appeared with gene therapy in mice is I think that we need a couple more papers like that over the next year, hopefully sooner, because we've got to answer some really important questions. What is the right vector to use? What is the right promoter to use? When is it too late in development, or what's the optimal time in development of when you should be giving gene replacement? And the big four is, and this is going to take a little while, I would really like to know if you found the perfect scenario for all of this. I want to know, what do those mice look like 6 months, 9 months, 12 months after getting gene therapy? Did it cause any problems? I think that's a big question, and part of that answer, you're going to go 12 months. But I think that's the sort of thing that we need to know about safety for a lot of those things that we talked about. So I think there's a lot more work that needs to be done. I hope, I've heard rumors that sort of thing is happening now, and the group the Gao et al. Group, I'm sure they're working on that next step in this whole process, which I think is really exciting. So we look forward to that. So I think we're still waiting.
Okay, so I have a question jumping off of the boys. The question is, have there been any trials that have only been for girls?
Only done girls?
Yeah.
Not that I know of. I'm trying to remember. I don't have my book.
Maybe? I don't know.
I'm pretty sure that boys were allowed in both trials, but I could be wrong.
I think so.
I don't have my book here to pull out the inclusion and exclusion criteria, but those are going to, if this is the note, the learning point here, you can go to clinicaltrials.gov and it will tell you the inclusion and exclusion criteria for every trial.
Okay, perfect. So, okay. So can you share any news about any protein replacement therapy in the pipeline?
I don't know anything about it.
Okay.
I think if I'd done the review of that study I did the review of that before.
In 2018?
Yeah, but that's a one swallow.
Okay. Okay, so guys, the one swallow, he did review what was going on, and it's recorded, so we can point you to that. Let's see, there are many timescales to when gene therapy will be available for clinical trial. Timeframes as little as 18 months. When do you think we might be going into clinical trials for gene therapy? And do you think we need to be more conscious about overexpression, as you highlighted, and safety issues?
Yes. I think we need to be more conscious about it. I think 18 months is very optimistic. I think we should all be half full. But I think that we also need to be diligent in asking those questions. Are we really sure that we've addressed that issue about overexpression?
Okay. Is it possible to find out the location of a gain, duplication, plus possibly mosaic mutation in a boy? And could this tell us more about long-term prognosis?
Katie.
I'm not sure I completely understand the question. So, doing genetic testing should determine where, if there's a duplication present. Most testing that we do these days will do both sequencing and deletion duplication analysis of the gene. So if there's a duplication present, it should be detected, and we should be able to determine approximately where the breakpoints are. So anytime we talk about a duplication, we have to be careful, because there's a duplication of the full gene that might also include other neighboring genes is one thing. A duplication of a base or a couple of bases, so a couple of letters in the code, is a very different type of mutation. So we're talking about two very different things there. But genetic testing should be able to determine if there's a large full gene duplication or if there's a small intragenic duplication. And then what was the second part of the question? Something about mosaicism?
Yes. Possibly. How would you know if your boy was mosaic is what I think she's asking.
Yeah.
Would that be reflected?
Mm-hmm. So if the genetic testing was done recently, if mosaicism was detected, most genetic testing reports are going to say possible mosaicism or suspected mosaicism. Most of them will not make a definitive claim that mosaicism is present, because anytime you're doing genetic testing, there's a certain level of artifact. So there's the sequencing technology isn't going to be perfect every time. So they need to be confident that if there is a change that's present in a proportion of the cells, which would be mosaicism, that that is true mosaicism in that patient and not just an artifact of the testing. So they're never going to report out a specific level. And even if they do, that doesn't really tell us very much, because it's not going to reflect the level of mosaicism that might be present within the brain. So the short answer to all of that is that a genetic testing report should tell you whether mosaicism was detected. But if it doesn't say mosaicism was detected, that doesn't necessarily mean that it's not there. It's just sort of the threshold of what a genetic testing lab would call, this is a mutation, it's present in all of the cells, versus we only see this mutation in a portion of the cells. So it's not perfect, but it's better than it was several years ago. So if this is outdated testing, if your child had testing done a while back, it might be worth doing it again if there's any suspicion for mosaicism, because current technology is going to be better at picking that up. And then in terms of what can be concluded from that, we can make general assumptions that a mosaic patient is typically going to be less severely affected. But again, because we don't know a precise level within the brain, we can't make generalizations, and we can't guarantee that a mosaic patient is going to be less affected than a patient who does not have mosaicism.
So is it fair to say that mosaicism is also, it takes a clinical, it's a clinical diagnosis as well? You guys play a big part in kind of telling people this is why we think your child presents this way?
I think we play a big part in kind of interpreting what mosaicism might mean for any one patient. So there's the genetic testing, there's the clinical diagnosis, and it's really putting the two together. One of them is not going to give you the full picture. It's important to have both.
So I would say that the translational field is still trying to understand more about is it bad to have 2 copies of CDKL5? So I think more studies need to be directed at that to understand, is that a problem? If so, how is it a problem, and why? You know, what is it doing something special to synapses, spines? What is it, so that we can understand that better? I mean, that's really important, because we need to know the answer to that with regards to gene therapy, because that's all about the promoter. You might put the gene in, but if you put a promoter in that doesn't regulate how much of the RNA that you're going to make, then you could make way too much. And if having too much CDKL5 is a problem, then you've created a problem.
Okay, so I think we might be approaching our last question here, and I think this will be an easy one for you. What process does a parent need to follow to qualify for clinical trials in the States if you are a non-US resident? Is there residing country that approves? Example, they're asking if they need permission to come to the US to do trials in the US. So can an international patient come to the US and participate in trials?
I don't think that I mean, that's a visa question. That is not.
There you go. Well,
It's a visa question.
just because, so because they live out of the country doesn't mean it would exclude them. It just really would be dependent on the study and the company, right?
And
Is that fair?
having said that, if you're going to participate in a trial, you almost have to come live here, because one of the aspects of trials in things like RET and CDKL5 are, we don't just want to know how well your seizures are improving. We want to know about everything. And so you need to be in an environment where you will thrive. So if you move for a trial, whether you live in Missouri and you're coming to Colorado or wherever, or vice versa, is participating in that trial still going to allow you to exist in your best state, where you have access to the therapies that are helpful and meaningful to you? So if being in a trial actually makes your access to therapies less than it normally would be, then that puts you in a situation where you would not be an ideal candidate for trial. And that is one of those questions that often is part of the inclusion criteria. Is participating in the trial going to interfere with your access to therapies?
Do you have time for one? Do you have time for one more?
I think so.
It's okay. I don't know if you have clinic. We're at the top of the hour. So I understand CDKL5 expression works as a modifying gene for other gene functions. Have we looked at which genes are modified, affected, and their metabolic disturbances?
So one of the papers that I didn't include was, there was a paper about how loss of CDKL5 affects function of mitochondria. And they're starting to look at some of those genes that are involved in mitochondrial function. And I think there's a lot of different studies and ways of pursuing this that we don't fully know about all the different genes that are turned off and turned on by CDKL5 yet. I mean, for MECP2, which CDKL5 may be interacting with MECP2, when you have loss of MECP2, you have hundreds of genes that are turned off, hundreds of genes that are turned on. It really is a lot of stuff to go through. And that's another reason the whole swallow idea is that, oh, hello, who's there in the background of Karen's thing there?
It's Samantha. Here she comes.
I thought that's who that was. Hello, Samantha. Anyway, so yeah, it's another aspect of why it's important to look at things with more than one swallow to see if they're really helpful or not.
Perfect. Thank you. So I want to be respectful of everyone's time. So I am going to turn off comments at this point, and we will be happy to follow up with families and help facilitate questions that arise after this. I'm going to mute, and I'm going to turn things back over to Karen, Heidi, and Tim.
Amanda saying hello to everyone. She's as disappointed as the rest of us that we're not in Houston right now. Tim, thank you so much. Katie, thank you as well. You did a fabulous job as always. Heidi, do you have anything to add?
You're on mute.
I was trying to share the article he was talking about. It didn't work out too well. Thank you both for joining us and sharing both the genetics and the recent research. I really love the how many swallows make a swallow a summer. And I think moving forward, that'll give some context to maintaining expectations.
I agree. Thank you for that example because it helps us understand how to look at different things and know just what you were saying. Is this something we can just translate to use it in our child or not? And that's very helpful. So for those of you still listening, our next session will be tomorrow. This is our last session for today. And our first session tomorrow will be at 11 a.m. Eastern time. That's also New York time. And we hope you will join us. So we'll be talking about some different databases you can participate in and be a part of to push research forward. Anyone else have anything else to add, or are we ready to say good evening? You're gonna wave too?
Thanks, guys, for having me.
Thank you.
Thank you.
Thank
Thank
you,
you.
everyone.
Thanks for having us. Good night.
Night.
Good night.